Humans have 23 pairs of chromosomes, and in each pair, one chromosome comes from the father and the other comes from the mother. What are our chromosomes made up of though? Our chromosomes are made up of DNA molecules. DNA is a polymer with two strands, and it is said to have a double helix structure due to it twisting. On these DNA molecules, there are small sections known as genes. Genes code for a particular sequence of amino acids to make a specific protein. These genes control your characteristics. One chromosome will contain many different genes which will determine your characteristics. The genome of an organism is the entire genetic material of that particular organism. The Human Genome Project wanted to find out as much as possible about the genome of different people. If the human genome can be understood, and it's not an easy task as each human genome contains tens of thousands of genes, it could lead to some great advances in medicine. Inherited disorders, disorders which are passed from the parents to the offspring, could be better understood. The genes which are linked to higher risks of certain diseases, such as diabetes, could be found. Also, the human genome could allow us to plan out the history of humans, such as family trees. Let's take a deeper look into the DNA structure. Each DNA strand is made up of thousands of nucleotides joined together. A nucleotide is made up of a sugar, a base and a phosphate. We say that the DNA molecule has a sugar-phosphate backbone. These bases attached to the sugar can alter, and there are 4 different bases, which are A, C, G and T. The bases are arranged in triplets, threes. Each triplet codes for an amino acid. It is in this way that a DNA molecule can determine the sequence of amino acids. Each gene will be made up of many triplet bases, each coding for a particular amino acid. If one of these bases changes, then a whole different protein could be formed. The rest of the video is now only higher tier content. In DNA molecules, there are complementary base pairs. These are pairs which are complementary to each other. In other words, in a DNA molecule, which is made up of 2 strands remember, the G and C will link up and the bases A and T will match up. This matching up of bases is important to how DNA functions. Protein synthesis is not a simple process. The first step is to produce a template for the protein of the DNA base sequence. The template will then leave the nucleus through a pore and bind to a ribosome. Carrier molecules are attached to specific amino acids. These carrier molecules will attach onto the template and allow the amino acids to come together. This process continues until a chain of amino acids is formed. The chain will then fold and form a specific shape. This is now a protein. The way in which the protein folds determines its use. For example, an enzyme will need an active site. Mutations can cause new genes to arise. Mutations only often appear small, such as one base in the sequence being altered, but this can actually change the whole functioning of a gene. Mutations occur continuously, however, and most of them don't actually alter the protein formed, or if they do, they only slightly change it, and so this means it will have the same function. Some mutations will lead to alterations. These alterations may mean that the protein folds in a different way, and this is particularly bad for enzymes as their active sites will change. Structural proteins could also lose some strength. There are many genes present on a single chromosome, and so all the genes are not always expressed. A gene is expressed when the protein it codes for is produced. A large proportion of DNA does not code for any proteins at all. Remember that genes are specific parts which code for proteins. The non-coding part of DNA has a role in switching genes on and off. They can also cause just parts of genes to be switched off, which again can change the protein that will be synthesized. This gene switching can change an organism's characteristics. This is also described as their phenotype. If a mutation occurs in the non-coding areas, then this gene switching on and off could be affected.